On October 7, 2019, Drs. Gregg Semenza, Peter Radcliffe, and William Kaelin were awarded the Nobel Prize in Physiology or Medicine for their work uncovering the role of hypoxic response in health and disease. Decades ago, each sought to answer the question of how kidney cells sense and respond to low oxygen levels, to ultimately stimulate the production of red blood cells and enhance oxygen delivery throughout the body. At the time, the nature and mechanism of hypoxic response was unknown and thought to be limited to a small subset of kidney cells. Exploring this basic science question, Dr. Semenza, Radcliffe, and Kaelin discovered that the hypoxic response is far more widespread than originally thought. Universally underlying core cellular processes and tissues and cell types. The transcription factor that regulates it all, called hypoxia-inducible factor, plays central roles in disease from kidney disease to cancer, diabetes, heart disease, stroke, and even immune regulation. These discoveries have opened up entirely new fields of research in medicine, with HIP-targeted treatments now yielding promising clinical trial results across a spectrum of diseases. Keystone Symposia is particularly vested in this story, as we have watched this emerging field unfold over the last 2 decades from a basic science exploration into a translational arena. Since 2004, our hypoxia meetings have provided a venue for these thought leaders to share their ideas and showcase their latest research, coming together with diverse academic, clinical, and industry audiences to discuss and direct the state of the field. It is only fitting that this year's meeting in January, which marks the 10th Keystone Symposia on Hypoxia, features all 3 Nobel laureates on the program. We are honored to host Dr. Semenza, Radcliffe, and Kaelin, along with their colleagues, for this meeting of the minds as the field of hypoxia takes center stage around the world. Here we catch up with each of these research leaders to discuss this journey from obscurity into the limelight and the future of this exciting field. So I guess I'd like to start out, obviously by saying congratulations. So what an exciting time.
So that's sort of the serendipity of science. You never know quite where things will lead. That's what makes it so exciting to do research. And in my case, it was studying the gene for a hormone called EPO, or erythropoietin, that controls red blood cell production and is regulated by specialized cells in the kidney that somehow can sense the oxygen levels, and if the tissue is not getting enough oxygen, signals for increased EPO production. And so we set out to first understand what controls where the gene is expressed in the body, and then after that to study how the expression of the gene is regulated according to the oxygen available.
Great. And as you discovered that it was much more widespread of a pathway than you thought, were there any particular moments or discoveries where you thought, wow, we're really onto something big here?
Yeah, so, we had first used when we wanted to understand the role of oxygen, we had gone from animal models to cell line and used a cell that had been previously shown to produce EPO when the cells were cultured under low oxygen conditions. And we knew that most other cell lines did not make EPO in response to hypoxia, but we asked, is HIF1 induced by hypoxia in those cells? And in every cell line that we looked at, when we cultured the cells under low oxygen conditions, HIF1 activity was induced. So that told us that it must have other targets besides EPO that accounted for its induction in every cell type. And so we thought about what are the kind of really critical responses to hypoxia that cells might want to activate. And one was the switch from oxidative to glycolytic metabolism, and we showed that genes for glycolytic enzymes were controlled by HIF1. And then we also showed that the production of angiogenic factors to stimulate new blood vessel formation, including vascular endothelial growth factor, were also controlled by HIF1. I decided that we should study the common diseases, the diseases that are most likely to cause morbidity and mortality in the population, including ischemic cardiovascular disease and cancer. So we tried to understand the role of HIF1 in the pathogenesis of those diseases.
And so now that you've discovered that it really is a core factor in those diseases, what therapeutic opportunities have come up, and how has this kind of changed the view of medicine as well as practice of medicine?
Right, so it's interesting because in the case of ischemic cardiovascular disease, the normal response that I described to you, which is to increase the production of molecules like VEGF to stimulate vascular response to ischemia, that response is impaired with aging and with diabetes. And so the normal induction of HIF1 does not occur. And so there we would like to augment the production of HIF1. And so we showed that we could use HIF1 alpha gene therapy to stimulate blood vessel formation in mouse models of ischemia. In the case of cancer, it's the opposite. It's the cancer cells that are hypoxic, and it's the cancer cells that respond to that hypoxia by inducing HIF1 and allowing these adaptations that are good for the cancer but bad for the patient. And so in that context, we'd like to inhibit HIF1 activity as part of a cancer therapy.
Okay. Can you describe a little bit more what role it plays in diabetes and metabolic disease?
We know that the combination of aging and diabetes equals limb ischemia, and when it becomes very severe, it can require amputation, which is pretty amazing when you think about all the advances we've had in medicine and science, that still every day people are having their leg amputated because there's no other available therapy.
Yeah.
We can show that we can recover the perfusion of the limb by using HIF1 alpha gene therapy.
Wow.
So we hope that these sorts of approaches may be beneficial, particularly in a disease like that where there are limited therapeutic options.
Mm-hmm. I'd like to touch a little bit on the immune system and how HIF is involved in either immune activation or suppression.
It's really complicated because there's HIF activity in many different cells of the immune system, and in some cases it's seemingly opposite in its effects. And so in terms of the immune system in cancer, we know that the cancer cells produce proteins that help them evade the immune system. Either hypoxia or chemotherapy induces the expression of multiple proteins that are cell surface molecules that allow the cancer cells to evade both adaptive and innate immunity. And we've shown that if we, treat with a HIF inhibitor, we can block those effects.
And are there certain cancers that would benefit more from the HIF-targeted therapy than others, or is it a general underlying process for all of them?
Yeah, so that's a great question, because sure, we can say that, HIF1 is induced and is active in most cancers, but that doesn't necessarily mean that a HIF inhibitor is going to add to the beneficial outcome. And unfortunately, for the most part, that is a answer that can only be derived empirically.
Where do you see the field heading in the next few years?
Again, my own bias, is that the applications, the clinical applications now, now that there are drugs that induce HIF activity that are being tested now in patients with anemia due to chronic kidney disease, and those drugs may be useful in other contexts besides anemia, such as ischemia. And then with the recent report of a small molecule HIF2 inhibitor having some activity in kidney cancer, that again is very exciting news, and proof of principle that HIF inhibition may be useful in cancer. So I really expect that there will be many more drugs targeting the HIF pathway in the next few years.
Dating back to 1977, you've been to nearly 20 conferences. What keeps you coming back? What do you enjoy about the conferences, and what do you get out of them in general?
So everyone looks forward to being able to interact with everyone in the field, in both scientific sessions, but also informally, and to really get a sense of what the field is like. It's really impressive, the quality of science that I found at every one of the Keystone meetings that I've attended. A lot of what we do is based on what we learn, from other people's work. And so to the extent that we can learn that faster by attending the meetings, rather than waiting for something to be published, that's really important. Because, one thing is always built on another. And so I think it's really important just to get the data out as quickly as possible, so people can use it to shape their work.
Great. Do you have any particularly memorable connections you've made or insights you've discovered while you were looking at those posters?
Yeah, we've cooked up lots of experiments late at night at Keystone meetings. So.
That's great to hear.
Yeah, it'll be interesting to see, but I think the attendance at the meetings has been quite good. I think it's one of the biggest Keystone meetings. So that's a kind of validation, right, of the field. And I don't see that changing anytime soon. So
That's true.
Just again, well done to the Keystone Symposia for their great work setting up these conferences. They all come off pretty flawlessly every year, and
we try our best.
and I look forward to seeing everyone at the meeting.